Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuhu. Well, students, today we are going to discuss about the law of the independent assortment. Basically, law of independent assortment. As the name is indicating that independent, it means that genes are not depending upon the genes. What is that? It means that genes are not depending upon the genes. Like, let's suppose when there's a time of segregation, when the genes pairs are going to segregate, let's suppose that these are the genes of the eyes, blue and the black eye. Let's suppose black and blue eyes. This is the gene of the eyes. When the time is going to come, they are going to segregate during the gamete formation. One gene pair will be in one gamete and other gene pair will be in other gamete. This used to happen during the segregation. But in this law, the Mendel have explained that the genes, the different genes, let's suppose the eye genes and let's suppose the height genes, they are not depending upon each other. They are not depending upon each other. They are going to segregate independently. They are going to do what? They are going to segregate independently. Let me to explain you in detail. It is like that, students. Look at here. It is like that. Okay, it is like that. Okay, let's suppose the person have the blue eyes. It doesn't mean a person who have blue eye will always have the white color. No, it is not like that a person who have the black eyes would have the brown hairs. It is not like that. It means that the blue eye color is not depending upon the white skin. It means that the black eye color gene is not depending upon the brown hair color genes. It means that they all are independent. When the time of the segregation comes there, when the genes are going to segregate, they are going to segregate independently. They are not going to depend upon each other. So this is known as the law of the independent assortment. So I'm going to explain you in very much detail that what do you mean by law of the independent assortment. So if you're going to study from here, now to study that law of the independent assortment, what the Mendel did, the Mendel done the experiment and he have done the dihybrid cross. What is a dihybrid? If you remember, I have told you about the monohybrid. Now this is what? This is a dihybrid. This is what? This is the dihybrid. So in the mono, if you remember, we were studying only one gene, but in that case, what, in the di we are going to study two genes at the same time. So the cross in which we are going to do the study the two genes will be known as a dihybrid. So in the next cross, Mendel studied two contrasting traits at the same time, and such crosses were known as a dihybrid crosses. Now he did what, student? It is very much interesting to know. He did what? He have chosen, he performed the experiment, and what he did, he basically have performed the experiment. Let me to see from here, sorry. Yeah, he have performed the experiment. In that case, he have done what? He have taken the two traits. One was the seed trait. It means he have chosen the two traits of the seeds. One was the shape of the seed and other was the color of the seed. The trait of the round seed controlled by the allele R was dominant over the wrinkled allele, that was the small r, and the seed, similarly yellow seed color controlled by the capital Y was dominant over the green controlled by the small y. So Mendel did what? Mendel did the cross in between the pure breeding of the pure round and the yellow seeds, plus he done the cross in between the two breeding of the wrinkle and the green seed. So let's study this cross. Okay, let me to explain you. It is like that, student. He did what? A Mendel had chosen the two traits of a seed. One was the color and one was basically the shape. If we are going to discuss about the shape of the seed, then there are basically two shapes of the seed. The seed shape is, if you are going to see from here, then it is very clear to you that one of the shape of the seed is wrinkled and other is the round. While the color, if you are going to see the seed, one is the yellow and other one is the green. So beta, if you are going to look at here, then this color is the dominant over the wrinkle seed. So wrinkle seed is basically recessive. This is what? This is the recessive. While this color yellow is the dominant and the green is the recessive. So he did what? He have given the sign of capital R for the round seed and the capital Y for the yellow seed. He have given the small r for the recessive seed and the G, or you can say small y, for the green seed. Okay, now this was the trade. Now what he did, he have done the cross. He have done what? He have done the cross in between yellow and the true breeding of the yellow and the round seed. He have done the cross in between the true breeding of the yellow and the round seed and the true breeding of the wrinkle and the green seed. Okay, he is going to do the cross in between these both. Now what happens, beta? This is basically known as a P1 generation. During the gamete formation, what will happen? During the gamete formation, the gametes are going to form, and what will happen, beta? These are gamete formation, beta. You have to be very much careful while making the gametes. It is like that. One, obviously there are two gene studies, so obviously in one gamete there would be two genes. Okay, capital R and the Y, capital R and then capital Y again. Now this capital R and capital Y, then this again capital R and capital Y. The four gametes are going to produce. Now in this case what will happen? Just state the gamete formation of this one. It is like that: small r, small y, this with this. Now this small r with this small y. So small r, small y. Now again this r with this. Sorry, the next one. This r with this y, small r, small y. And now this r with this y. So small r, small y. So the four gametes are going to produce, right? Now let's study in the during the Punnett square what is going to form. If we are going to study in the Punnett square, what is going to form? One gametes on one side, one parent's gametes on one side, if you remember. Other gametes on other side, if you remember. Okay, now students, do form its gamete and what would be the result? It is like that: capital R, small r, capital Y, small y. Then again capital R, capital Y, small y. Then again capital R, capital Y, small y. Then again capital R, capital Y, small y. Okay? So it is like that: capital R, capital Y, small y. Okay, this is the result. You can see from in the next class, in the next cross as well: capital R, capital Y, small y, capital R, capital Y, small y, capital R, capital Y, small I. So it is like that. The resultant is this one. In the F1 generation, what should we get? In the F1 generation, the result is capital R, capital Y, small y. So Mendel have observed that 100% in F1 generation, the 100% seeds were round and yellow. What he did, obviously because the round is dominant over the wrinkled and the yellow is dominant over the green. So what was the phenotype? The 100% round and the yellow seed. Now Mendel did what? Mendel did the cross in between F1 generation and the F1 generation. Now what will be the result? Let's see. So students, if you are going to look from here, if you are going to look from your book, it is like that. Okay, this is a round yellow plant and this is a wrinkled green seed color. Fine. In the F1 generation, in the P1 generation, what he did, the Mendel have done the cross in between the true breeding of the capital R and Y, and the true breeding of the wrinkled and the green seed. And in F1 generation, what he get? He get all round and all yellow. I have explained to you before. Now we are going to move in the next generation, that is F1 and F1 generation. What he did now? he have done the cross in between the F1 and F1 generation. So F1 generation was the capital R, small y r, capital Y in the small y. That means the round seed with the yellow color. The cross between the wrinkled seed, round round seed and the yellow seed. So he have done the cross in between. So first step is obviously, student, what is this first step? That is a gamete formation. So when the gametes are going to form, I have told you how the gametes are going to form. R and Y. Obviously that there are two genes, so each gamete will contain the both gene. So R and Y. Similarly this R and again Y. R in the small y. Now this with this one, small r and capital Y. Okay, small r and the small y now. Now in the same case here, gamete formation, capital R, capital Y, capital R in the small y, small r in the capital Y, small r and the Y. Now we are going to make the Punnett square, in which we are going to see the what would be the result. So one parents on the one side and other parents gametes on the other side. So if ry in this one. Now similarly in that case, capital R, small y, capital Y, capital Y R, small y, small r, capital Y, and the small r, small y. Now let's draw the cross in between. So it is very simple, you know it very well. Capital R, capital Y, capital Y, capital R, capital Y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. Similarly in that case, capital R, capital Y, small y, capital R, small y, capital R, small r, capital Y, small y. Again look at here, capital R, small r, capital Y, small y. Again, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y. ठीक है, capital R, small r, capital Y. Then again, small r, capital Y, small y. Now the last one, capital R, small r, capital Y, small y, capital R, small r, small y, small y. Now again here, capital R, small y, small r, capital Y, small y, small r, small y, small y. So this is very beautiful cross. Now let's see what is inside there. If you are able to read the genotypes, then you are able to tell me the phenotype. This is a round and the yellow. So this, let's see that how many plants were the round and yellow. Let's see, count that. Round and yellow 1, round and yellow 2, round and yellow 3, round and yellow 4, round and yellow 5, round and yellow 6, round and yellow 7, round and yellow 8, round and yellow 9. So 9 were basically round and yellow. Now let's see the next trait. This is what? Round and green. Okay? Round and green. So let's see how many are the round and green. Okay, this is round and green. This is round and green. 2, 3, round and green. So 3 are basically round and green. Let's see the next trait. Wrinkled and yellow. Wrinkled and yellow. So let's see that trait. This one is a wrinkled and yellow 1, wrinkled and yellow 2, wrinkled and yellow 3. So 3 are the wrinkled and yellow. Now this one is what, beta? This is the wrinkle and green. So this means that 1. So what's the ratio in between them? What is the ratio? There is a 3 ratio, 9 ratio, 3 ratio, 3 ratio 1. So now what is proved by this experiment? What is proved by this experiment? Student, in short, it is basically said that all the genes are independent to a sort. It means that the yellow color were not depending upon the round seed. Similarly, you can also say that the yellow this we have get the different traits. These were the different traits, fine, and this is the ratio which we obtained, right? 9 ratio, 3 ratio, 3 ratio 1. So what is actually meaning of this? The meaning of this is beta. The meaning of this is very simple. It means that the the round is not depending upon the yellow color. Similarly, the round seed is not depending upon the yellow color. The green seed, the green color is not depending upon the shape. This means that the shape color and the shape of the seed and the color of the seed, they are not depending upon each other. They are independent to assortment. That's why we have obtained that much variety. We have obtained too much varieties of the plant. It means that they are allowed to assort independent. It means that nothing is depending upon each other. It is not like that all those who have the white color are going to be have the high IQ level. It is not like that all those have the high IQ level, they are going to have the blue eyes. No, it is not like that. It is not like that all who have the black hair will have the black eyes. No. What is actually mean of this? This means that all the genes assort independently. They are not depending upon each other. Clear? Hope so you get that. So let's go through that law. It is very much important law. That's why students I'm explaining you again and again. It is like that. Let's see the summary. The Mendel have done the dihybrid cross, and he have done the experiment in which he have chosen the seed trait. One was the shape and other was the color. What he have done in that trait, he knows that the shape of the round shape is dominant over the wrinkle shape, and the yellow color is dominant over the green. Fine. Now what is next? What is next now? What he did next? He had done the cross in between, and first he have taken the parental generation, and the result of the F1 generation was that all the round and the yellow, all the seeds were the round and yellow. Then he have done the cross in between F and F1, and then there was the gamete formation, and then in F2 generation what happens? In F2 generation, you can see here it is all clear. In F2 generation what happens? 1 2 3 4 5 6 7 8 9. Hold on students. It is like that, yes. 9 were what? 9 were the round and yellow. Okay. Then there was a 3 1 2 3. 3 were the wrinkled and yellow. Then there was 3 were the round and green. 3 were the round and green, and 1 were the wrinkled and green. So this was the ratio. So what he get from this ratio? He get what? Mendel explained that the two traits, seed shape and the seed color, are not tied with each other. Like they are not tightly bound with each other. The segregation of the R and the Y will, what will happen? R and the Y will what? It will be what, student? It will be independent of segregation. Capital Y and the small y alleles. From his second experiment, Mendel concluded that different traits are inherited independently from one another, and this principle is known as the law of independent assortment. Hope so you get that. If you have any query, you can ask me better. Do the practice of the cross. This is too much important cross, and this question used to come mostly in the paper. So best of luck and take good care of yourself. And I want all of you to see that video.